introduction. Hello, everybody. I'm Danny Wu, and I'm a PhD student from Johns Hopkins University. And today I'm happy to be here to talk about our recent studies on the longitudinal changes in cerebral metabolic rate of oxygen in older adults without and with cognitive impairment. We know that our brain is about 2% of the total body weight, but consumes about 20% of the total energy. And the energy the brain consumes mainly comes from the oxygen that are extracted from the blood. Here, the cartoon here shows that how our brain tissue extracts the oxygen from the blood. The blood in the arterial side is almost fully oxygenated, and it is delivered to the capillary and finally drained to the vein. At the interface between the capillary and the brain tissue, a portion of the oxygen that are carried by the blood is extracted by the brain tissue and finally consumed by the neurons to support neural activities. This brain oxygen could be described as a mathematical relationship shown here. So in this relationship, we have CBF representing cerebral blood flow, and it denotes how much arterial blood is delivered to the brain. And we also have OEF representing oxygen extraction fraction. It is the difference between arterial oxygenation and venous oxygenation. And also we have CMRO2 representing cerebral metabolic rate of oxygen. It denotes how much oxygen is extracted by the brain tissue. So actually CMRO2 reflects the oxygen consumption of the brain, and CBF reflects the oxygen supply or the blood supply of the brain. And OEF actually reflects the balance between CMRO2 and CBF. That means it reflects the balance between the oxygen consumption and oxygen supply. These three physiological parameters could be used to study the brain metabolism. And there are some interesting findings in the previous studies about how these three parameters changes during normal aging and diseases. So during normal aging, it was found that CBF will decrease with age and OEF will increase with age. But for CMRO2, the results are mixed. Some reports that CMRO2 will increase with age, but some reports that CMRO2 will decrease or remain stable when people get older. And for diseases, it was found that CBF was lower in patients with small vessel diseases, and CMRO2 was lower in patients with cognitive impairment. Because CMRO2 reflects the oxygen consumption, so it might relate it to the neural function, and so CMRO2 might be lower in people with like neurodegeneration. And as we talked before, OEF is a balance between CBF and CMRO2. So for people with vascular risk, OEF will increase, but for people with cognitive impairment, OEF will decrease. Most of the previous studies use cross-sectional data. And for our study, we want to use the longitudinal data to further investigate the effect of the aging and diseases on these physiological parameters. So first, let's take a look at how we measure these physiological parameters non-invasively using MRI. We use the TRUST MRI to measure the OEF, and we use phase contrast MRI to measure the CBF. And CMRO2 is calculated from CBF, OEF, and YA, which means the arterial oxygenation, and also CH, which means the oxygen carrier capacity of the blood. So our study, the participants in our study were part of the BioCard cohort. The participants in the BioCard cohort received comprehensive cognitive assessment, clinical assessment, and MRI scans, as well as blood biofluid sampling. In our study, we include 174 cognitively normal participants and 55 participants with cognitive impairment. Their age ranged from 50 to 92 years old, and each participant will receive the MRI scan every 2 years, and the MR scans they received range from 1 to 4 times. So in total, we got 544 MRI measurements. So let's first take a look at how what we measured from a technical point of view. So here, this plot shows all the OEF and the CBF we measured by using the MRI. And we found that there is a strong correlation between the OEF and the CBF. But we know that there exists physiological noise during the MRI measurement. The physiological noise might come from, let's say, breathing pattern or drinking coffee. But theoretically, the OEF and CBF is negatively associated with each other, according to the mathematical equation. But in our real measurement, we found that the OEF and CBF we measured is still in a negative correlation. And since we calculate the CMRO2 from the product of CBF and OEF, so we found that we could see that the CMRO2 we calculate for the CMRO2 we calculate the physiological noise will be canceled out, and we use this measurements to do the statistical analysis. And for the statistical analysis, we use the linear mixed effect model to in all of our analysis. And so first, we investigate the age effect of the physiological parameters in the physiological using the linear mixed model. And for the independent variables, we include sex, education, time since baseline, and age at baseline. So here, time since baseline means the follow-up time. For example, if a patient received MRI scan 2 years after his first MRI scan, then the time will be 2 years. So first, for CMRO2, we found that male has a lower CMRO2 than female. And we also found that CMRO2 increased with time longitudinally. That shows us that when people get older, the brain is working harder to compensate for its lower efficiency. So it extracts more oxygen from the blood. But we found that the CMRO2 also decreased with age cross-sectionally. That might show that the brain might run out of compensatory capacity and shows the signs of neurodegeneration. We think these two results are not contradictory to each other because it actually evaluates the CMRO2 change at different time span. So here for the longitudinal time, it's 2 to 6 years. But for the baseline age, it's 50 to 90 years old. So we further investigate the slope of CMRO2 change with time. And we found that the slope of CMRO2 change with time is negatively related to the baseline age. That is to say, if when people get older, their CMRO2 will start decrease. The figure at the right-hand side also shows the same meaning. Like here, one line corresponds to one individual, and the slope of this line is the slope of the CMRO2 change with time, and their baseline CMRO2 are corrected to 0. So from this plot, we could see that when people get older, their CMRO2 will increase in a slower rate and even start to decrease. For OEF, we found that OEF will increase with time longitudinally. And for CBF, we found that CBF will decrease with baseline age cross-sectionally. And we also found that male has lower CBF than female, which is consistent with the previous studies. And we did 55 participants with cognitive impairment into our analysis. So here we add clinical diagnosis as an independent variable here. So for the diagnosis, people with cognitively normal people were coded as 0. And the people with cognitive impairment, which might be MCI or dementia, they are coded as 1. And from the result, we could see that CMRO2 was lower in cognitively impaired participants. That might suggest that CMRO2 is a sensitive biomarker for neurodegenerative diseases. We further investigated the ApoE4 effect on these physiological parameters. ApoE4 is a genetic risk factor for the late onset Alzheimer's diseases. So when people have more ApoE4 alleles, that means he will have more risk to develop to the Alzheimer's disease. And we found that if people have more ApoE4 alleles, their OEF will be lower. That is consistent with the previous study. And we also investigate the difference between the two subgroups. And we found that people with 2 ApoE4 alleles will have lower OEF compared to non-carriers. And for CBF, we also found that people with 2 ApoE4 alleles will have a higher CBF compared to the non-carriers. So these results tells us that the brain physiological alteration is actually is more pronounced by the homozygous, which is the 2 ApoE4 alleles. And from the mathematical relation here, we could also conclude that the decrease of OEF that is shown in the previous study is actually caused by the increase of CBF, with CMRO2 remains the same because we didn't find any significant results between CMRO2 and ApoE4. So to summarize, in this study, we found that there was a complex trajectory of CMRO2 during aging in cognitively normal adults. And we also found that CMRO2 was lower in participants with cognitive impairment. So it might be a sensitive biomarker for neurodegenerative diseases. And for the genetic risk factor ApoE4, it had no effect on CMRO2, but was associated with CBF and OEF. The brain physiological alterations by ApoE4 were most pronounced in the homozygous, which is the 2 ApoE4 alleles. Thank you.